Chapter 14: Rhizosphere Chemistry in Relation to Plant Nutrition. The outline of this presentation will be spatial and temporal characteristics, rhizodeposition and root exudates, and rhizosphere pH and nutrients. Here's a quick look at the variability in soil characteristics that can exist within just a few millimeters. Plant roots have adapted to manipulate the chemical and biological attributes of their immediate soil surroundings. The characteristics of the rhizosphere can vary both spatially and temporally. This variability is determined by the radial and longitudinal root axis, as well as diurnal variations. Radially extending outwards from the root's vascular tissue is the endorhizosphere. This area extends from the apoplasm of the root rhizodermis to the outer root cortex. The primary soil-root interface on the outer surface of the rhizodermis is the rhizoplane, and just beyond this point is the ectorhizosphere. Collectively, these regions make up what is referred to as the rhizosphere. The radial extent of this realm can vary from less than 1 nanometer to several centimeters. Nutrient concentrations within this region can vary as determined by a compound's capacity to diffuse. Nutrients that are found in less concentration in the soil and lack mobility, such as phosphorus, potassium, ammonium, and micronutrients, are quickly taken up by the plants. Therefore, they are found in less concentration in the rhizosphere. Compounds comprised of more soluble elements, such as calcium and magnesium, are more prevalent in soils and therefore can accumulate in the rhizosphere. Above all, the ability for a plant to uptake certain nutrients is highly regulated by the electrochemical gradient and the bulk soil nutrient concentrations. Image C illustrates how excess calcium in the form of calcium carbonate can accumulate in the rhizosphere. This can lead to precipitation within the root structures and cause damage to the root's cellular integrity. The graph to the right, adapted from Hissinger 2004 and Junk 2002, illustrates the rapid uptake of certain nutrients at the root surface, attributing the longitudinal gradient of the rhizosphere is highly varied in both chemical and biological characteristics. Generally classified by two regions, the meristemic region of active growth, either in the form of elongation or root hair development, is known as the apical root zone. This vicinity is characterized by its ability to uptake water and nutrients due to its lack of endo and exodermis, as well as its larger surface area. The mature region closer to the base of the plant is known as the basal root zone. This section has a higher microbial population because of its lack of mucilage, which is generally attributed to preventing higher concentrations of outwardly diffusing microbial stimulants. Table 14.2 from Marschner 2011 offers a helpful schematic comparing the chemical and biological differences between the radial and longitudinal gradient. The chemical behavior of roots can also be determined temporally. The figure here from Tagaki and Newman demonstrates how at different periods of the day, barley roots secrete a chelating compound in order to increase iron acquisition. Similar behavior has been documented in other species under phosphorus limitation as well. It has been thought that such mobilization efforts are influenced by certain developmental stages. While the spatial and temporal characteristics of roots are crucial in understanding rhizosphere chemistry, the substances released from roots is the driving force behind rhizosphere's unique chemical environment. These substances are categorized as rhizodeposition, comprised of sloughed-off root cells and tissues, and root exudates, which are further subdivided into three categories: diffusates, secretions, and excretions. The figure above, adapted from Newman 2007, illustrates the subdivisions of different forms of rhizodeposition. Of the total photosynthetic carbon that a plant synthesizes, 20 to 60%, depending on the species, is allocated below ground. Not only does this play a crucial role in plant health and microbiome dynamics, but these substances account for 30 to 40% of all organic matter input in soil. A byproduct of normal root growth and development, root cells and tissues are released into the rhizosphere. Cells that are passively released during root turnover or damage are referred to as lysates. Cells that derive from the calyptra and undergo a controlled release are known as border cells. The hormones auxin and ethylene regulate growth processes, but this form of rhizodeposition can also be stimulated by environmental factors such as water, carbon dioxide, or mechanical intrusion. Shedding is achieved through the release of pectolytic enzymes that hydrolyze the cell wall pectin. This action produces hydrogen ions, which further assist in the degradation process. Cells released are then embedded in the mucilage and are sent upwards towards the basal region. As the cells move upward, they attract potential pathogens and microbes away from the new vulnerable apical growth. Sloughed-off root cells and tissues can also contain antibiotics, further serving as protection to the new growth. These compounds have also been noted for their ability to chelate with aluminum and detoxify the rhizosphere. Organic compounds released into the rhizosphere are referred to as exudates. Based on their mode of release and chemical composition, they are further subdivided. Diffusates are low molecular weight compounds passively released into the rhizosphere. The majority of diffusates are sugars, phenolics, amino and organic acids, most of which are immobilized due to rapid microbial consumption. But there is an active reuptake mechanism in place that is signaled when plants are nitrogen or carbon stressed. High molecular weight compounds that are actively released into the rhizosphere for a specific function are referred to as secretions. Mucilage, a gelatinous polygalacturonic acid polysaccharide, protects against toxins such as aluminum and heavy metals. This substance also improves soil structure by aggregating particles. The resulting substance is referred to as mucigel, which is a mixture of mucilage, microorganisms, and soil. This product improves nutrient retention, especially for phosphorus and micronutrients, by adhering soil particles to the root surface, increasing the proximity and ability to uptake. Mucilage has also been associated with assisting in the desorption of nutrients from clay particles. The secretion of ectoproteins can also aid in nutrient mobilization. In soils with limited phosphorus, the release of phosphatase induces mobilization and uptake by plant roots. Other ectoproteins such as chitinase, glucanase, peroxidase, and phenyloxidase were secreted during defense reactions. Additionally, proteins can help initiate symbiotic relationships with soil microbes. For example, the enzyme invertase is associated with establishing mycorrhizal, fungi, and plant interactions. Excretions are comprised of low molecular weight compounds deposited under controlled release. These substances include metabolic waste products and toxins that have accumulated in the plant system. Some examples are lactate under anaerobic conditions, malate and citrate during bicarbonate toxicity, and carboxylates associated with aluminum toxicity. Excreted substances are also associated with the mechanisms enlisted by nutrient-stressed plants to mobilize availability of nutrients such as zinc, phosphorus, and iron. Important symbiotic relationships with nitrogen-fixing microbes and mycorrhizal fungi are also dependent on the excretions of phenolics, flavonoids, and strigolactones excreted into the rhizosphere. Many rhizosphere relationships and chemical interactions are dependent on the pH within the soil-root interface. Modification of the rhizosphere pH is also keyed to nutrient availability and mobilization. pH determines nutrient solubility within the soil solution. Therefore, it is essential to nutrient availability and potential toxicity to plants. The pH within the rhizosphere is highly variable depending on location and nutrient demands. Along the root axis, the apical region is characteristically acidic, while the basal zone is more alkaline. In order to create the electrochemical gradient required for nutrient uptake and maintenance of its own internal pH, plants release protons into the rhizosphere. This acidification adds to the chemical weathering of minerals, assisting in the mobilization of nutrients like phosphorus and manganese. In some instances, such as the uptake of nitrate and other negatively charged particles, roots can alkaline the rhizosphere with the release of hydroxide. Under more severe alkaline conditions, essential micronutrients become insoluble and thus unavailable to plants. Certain species in the Gramineaceae family, known as strategy 1 plants, acidify the rhizosphere as a mechanism in coordination with the PEPC pathway, aiding in the mobilization of uptake of iron. Thanks for listening to my lecture. Here are a few of my exam questions. Which of the following statements about the basal and apical region is true in regard to rhizosphere characteristics? How do rhizodeposition and root exudates differ? Which of the following nutrients could be made more plant available as a result of root exudation? What characteristic of the rhizosphere is highly dependent on pH status?